So what are students actually going to be doing in regards to different procedures to help you learn the different events that happen in mitosis? Well, the first thing that we have are models that are available for students to look at. The models, we have two separate sets. We have a set of animal cell models, and we have a set of plant cell models. So one of the things we want students to do is to take some time and examine these models. Look at the cell shape. Look at the nucleus. So for example, you can see that even in the smaller picture that the nucleus still exists. You can see that the nucleus is starting to break down. You can see by the time we get to metaphase here, the nucleus is completely gone. By the time we get to the end, you can see that two nuclei have been reformed. We want students to look at the chromosomes. Are the chromosomes in the duplicated state? Do they consist of sister chromatids, or are the chromosomes in the unduplicated state? Are they sort of full-fledged chromosomes? We also want you to take note of the differences in regards to what we talked about with cytokinesis. So for example, in this plant cell here, you can actually see the little cell plate. Those would be those little vesicles with cell wall materials that are accumulating sort of along the metaphase plate, sort of the center of the cell. And you can see that when the cells are completely formed, that cell plate actually becomes a cell wall, and it actually stretches across the entire two cells, so from one side to the other. So again, take some time, familiarize yourself with these different models so that you can recognize these different stages if you are shown the models. Now, if you are in lab, we do have beads that can be used to help simulate the stages of mitosis. And what we want students to do is, as they simulate, we want you to draw out the different events that are happening in prophase. And we also want you to take time to write the key events. So for example, we talked about what happens in prophase. For example, that the nuclear envelope is beginning to break down, that the mitotic spindle begins to form, right? That's important because it's going to move the chromosomes around. You can also draw in or mention here centrosomes. So these little lines are really there to help provide a space for students to really focus in and key in on the major events. Things that we want students to be able to do are not only recognize the different stages, talk about the differences, for example, between plant and animal, but we want you to be able to discuss what's happening in each phase and why that needs to happen in order to allow for the cell to divide. Now, in procedure 9.2, we are going to have students observing mitosis in plant cells. And to do this, we're actually going to use a root tip from an onion. So we're going to use allium. Now, what you see here is sort of all of the stages of mitosis in plant cells. So some of these cells are going to contain condensed chromosomes, which have been stained so that you can more easily see them. So for example, we can see a cell that's in metaphase here. This little cell up here is in anaphase. This little cell over here looks like it's in prophase. So this image comes directly from your book, and you should become familiar with what these different stages look like. We also want you to observe the stages using specific images that we provide. So again, these images were taken from your lab book, and if you are shown these images on the lab quiz or the lab exam, we want you to be able to identify them. We want you to be able to say, oh yeah, that's telophase. I can kind of see the two little nuclei. Oh, that's anaphase. I can see that the chromosomes have been separated. Here's metaphase. The chromosomes lying on the metaphase plate. This is just a staining artifact, so please don't become, at least that's what I think it is. It's a staining artifact, so please don't become distracted by that. So again, procedure 9.2 just gives students an opportunity to familiarize yourself with the different stages as they are represented in the different stages of mitosis as they're represented in plant cells. Procedure 9.3 is going to have students determine the amount of time spent in each stage during cell division. So the cell cycle, the length of the cell cycle is going to vary depending on the type of cell. Okay, so different cells, their cell cycles are different. They don't all last exactly the same amount of time. Some cells, such as the cells that make up your skin, go through the cell cycle often. Other cells, such as I mentioned earlier, your neurons, they are amitotic. They don't divide at all once you reach maturity. A normal animal or plant cell typically takes 24 hours on average to go through the entire cell cycle. But one of the questions that we often get asked is, well, how much time do cells spend in each of the individual stages? So how much time does the cell spend in interphase or prophase or prometaphase, metaphase, anaphase, telophase? We can actually figure this out. So normally if you're in lab, you would actually be using a microscope and slide, and you are able to actually count up in one field of view the number of cells in the different stages. So for the sake of this presentation, I've just made up some general numbers. So let's say that I'm looking through the microscope and I see this. What I'm really doing is I'm counting all of the little individual cells, and I'm basically going through and I'm determining, oh, this cell's in interphase. So this would get a little tally mark. Oh, this cell's in prophase. This gets a little tally mark. Oh, this cell's in prometaphase. This gets a little tally mark. And I basically am going to go through all of the cells in my field of view at 40x, and I'm going to account for all of those cells. So here's my individual data that I accumulated, that I actually counted. And then typically what we do is, in order to get a bigger sample size to reduce error, right? So the larger the sample size, the less error we expect, the less bias we expect, right? Big sample size is a good thing. What we do is we collate the data. So we take data from each individual student and we put that all together. So in this column, you can see that these are the class total numbers, right? So when we go to actually figure out how much time the cells are, or the plant is spending in each of these phases, there's a relatively straightforward little equation that we can use. So that little equation is hours of a stage X, whatever stage that is, you get to choose depending on what you're looking for, is equal to 24 times the number of cells in that stage divided by the total number of cells counted. So for example, if we want to know how much time cells are spending going through interphase, what we would do is take 3,492, because that's what was counted by you and your classmates, multiply that by 24, and divide that by the total number of cells counted. So this 4,324 represents these numbers all summed together. What that comes out to be is about 19 hours, 19.38 hours. When you do this for each of the phases, they should come up to approximately 24 hours. And what this is going to hopefully show you is that most of the time the plant is going to spend, really the cells are in interphase. Organisms don't divide just to divide. They divide when they need to, or when they're chemically cued to, for different reasons. Again, whether it's growth and development, whether it's repair. But it's not, cell division is not just a process that just happens. Actually, if we lose control of cell division, it can cause some very, very big problems. Now, the last thing that we have students do, the last procedure that we have students do in regards to helping you master and learn about mitosis is procedure 9.4. And in procedure 9.4, students are going to observe mitosis in animal cells. And we are going to use the fish blastula to identify the different stages. So again, these images that I'm showing you right here can all be found in your book. So this is a collection of several of these cells, all taken in one field of view. So we can see here, this would be telophase. Over here we have anaphase. Again, down here we have metaphase. This is just, I believe, a little staining artifact here. But then we have the individual cells broken down so that you can see each of the stages and you can compare and contrast. So in conclusion, what students need to be doing in the mitosis portion of this lab is really to learn to identify the different stages of mitosis using both the plant and animal cell models, to learn to identify the different stages of mitosis using plant cells, the allium slides, the onion root tip, as well as the whitefish blastula, the animal cells. It's important that not only can you recognize and identify the different stages when you're looking at these models or at these different cells in regards to slides, but that you can also talk about the important events that are happening in each of the stages. When does, for example, the nucleus begin to break down? When do the sister chromatids get pulled apart? Really, you will be held accountable for understanding and knowing when those different events happen. And it's also important that you can determine the amount of time spent in each of the stages during cell division. So knowing that equation and being able to calculate, using that calculation, the time spent.